Okay. So, okay. So we are going to talk about primarily special senses. The lecture itself is called special senses, but in reality, it also is all senses. But the only slide, but not about special senses, is this one, and so it's really a special senses lecture. But all of the sensors rely on sensory receptors. These are cells or proteins on cells that are designed specifically to sense something from our environment. And we have lots of different senses. And so we have a lot of different types of sensory receptors. We have the special senses, and then we have the somatic senses. Somatic senses are the senses that are not part of the special senses. And if you break down the somatic senses, we like to group them. Sorry, just a second. I'll be right back. There was a tick on my knee. I'll be back. Okay, so but that's a first for me, and I'm probably a first for you. I'd look down and I said, "What's that spot on my knee?" It's a tick. Okay, he's gone. Hopefully, I don't have line. Hopefully, I calmly says, "There's a tick on my knee." Yeah, like there's a snake in my boot. There's a tick on my knee, right? So, what are these people gonna think when they watch this video recorded semesters from now? They're gonna think I'm insane, which they're probably gonna be right. Anyways, now I'm kind of paranoid and I'm looking to see if there's any more. Okay, so somatic senses we break down into cutaneous and visceral senses. Cutaneous refers to skin. And so these are a lot of the senses that we think about with pressure. So how hard am I pushing on something? Am I touching something, or is something touching me? The pain, the temperature, things that you really probably associate primarily with your hands or your fingers. But we also have visceral sensory receptors. These are not things that you are going to be aware of. These are going to be connected to the autonomic nervous system. And so you can't respond to their signals. And so there's no reason for your brain to consciously tell you what these signals are. And so these include the baroreceptors and osmoreceptors. And you probably have no idea what either of these are. So baroreceptors, well, actually, before I tell you, does anybody know what baroreceptors are? Pressure, yes. So what part of that word, baroreceptor, reminds you of pressure? Yeah, exactly. The baro, like in barometer. Barometer is barometer, right? It's a meter that measures baro. I don't remember where exactly baro comes from, whether it's Latin or Greek or whatever it is. But a baroreceptor measures pressure. And in our body, it is measuring blood pressure. And so imagine you have, well, we're going to be measuring blood pressure in a blood vessel, right? So that's basically like a hose. Imagine you have a hose, and if you put high pressure into that hose, the hose is going to want to stretch and get bigger. Same thing happens with our blood vessels. When the blood pressure gets higher, the blood vessels stretch. And so what we do is we simply have receptors that measure how stretched our blood vessels are. Because if they're embedded in the wall of the blood vessel, when the blood vessel stretches, the baroreceptors stretch also. So that is going to give feedback to our central nervous system, telling it what is our blood pressure. Because if the baroreceptors are saying we have high blood pressure, but we're in a situation that we don't want blood pressure, or high blood pressure, we're going to do something to relieve that pressure. Probably we're going to reduce our heart rate. Now this is probably the harder one. Does anyone know what osmoreceptors measure?
Does it have to do with osmosis?
Close. So osmosis, yeah. So osmosis, remember, is a movement of water from one place to another based on solute concentration. So in this case, the osmo refers to osmolarity. And the osmolarity is a chemistry term that we are not learning. But like someone said, it is talking about concentration. Osmoreceptors are going to measure the concentration of stuff in our blood. Now this gives us a number of different ways to respond to that. If we measure that there is a lot of stuff, a high concentration of stuff in our blood, we can do a couple of different things. If we have a normal or concentration that we want to keep our blood at, imagine a situation where these osmoreceptors are measuring a very high concentration, higher concentration of stuff dissolved in our blood than we want. What are two ways that we can go about normalizing that concentration? So imagine it's salt water. If you have a specific concentration of salt water that you want, and somebody gives you a glass, and it is containing salt water that's too concentrated. There's two ways that you can do that you can fix it. What could you do? Add more water, dilute it. One of the things that happens when osmoreceptors detect high concentrations is they send signal to the brain. The brain says, I need to dilute this. And so if you're going to dilute your blood, what do you think your mind wants to do?
Drink more water.
Yeah, it makes you thirsty. Well, you get thirsty because your osmoreceptors are telling your brain that your blood is too concentrated. Now, what is a different way to solve that same problem? If you can't add more water, what do you need to do? Remove salt. Exactly. And so if you have the ratio from salt to water is too high, either add more water or reduce the salt. And so how do you think your body is going to remove salt? I heard you say it. I couldn't understand what you said.
Potassium?
No, simply, if you want, how do you get salt out of your body, do you think?
You urinate?
Urinate it, yeah. And so if you eat a bunch of salty food, in the short term, it is going to cause you to retain a bunch of water with that. But as you retain that water weight, you increase your blood pressure, which causes the osmoreceptors to send the signal to, or the baroreceptors to send the signal to your brain telling you that our blood pressure is high and the osmoreceptors are saying we have too much salt, and so we can kill two birds with one stone if we just urinate the salt out, and the extra water will go with it. So that is our very, very brief talk on somatic senses. The rest are going to be on our special senses. We're primarily going to focus on the eyes and the ears. I think we have 1 or 2 slides each on taste and smell. So we're going to start with taste. And the technical term for taste is gustation, but that's not something you're going to be tested on. So we know that we taste with our tongues, and one thing that you probably don't know or think you know but are actually wrong is when you look at your tongue and you see the bumps on your tongue, what do you call them? So basically everybody would call them taste buds. That is wrong. Taste buds are microscopic. What you're seeing when you look at your tongue are called papilla. They're little bumps, and on those bumps are taste buds. And taste buds are cells, individual cells. So now clearly cells aren't big enough that you can see them on your tongue. And so we have the bumps on our tongue that contain taste buds. Now, why do you think, if the bumps on our tongue are not the taste buds, why do we have bumps on our tongue? What's the purpose of them? I can think of 2 good reasons for those bumps to be there. Increased surface area. Yes, that's one of them. What is the benefit of increasing surface area on your tongue?
More taste.
Yeah. The more bumps that you have, the more room you have to cram the taste buds in there. If you have a smooth surface, you can only fit as many as you can fit. But once you add this, you add a whole bunch of surface area so you can add more taste buds. What is another benefit that you can think of for the bumps on your tongue? It has nothing to do with taste. Yes, that is exactly what I was thinking. It makes it a rough surface to grab on to your food. You don't think about it when you're chewing until you bite your tongue. But when you're chewing your food, you are constantly moving that food around, pushing it into the teeth every time you chew. And every once in a while, it doesn't get out of the way fast enough, right? And you chomp it, and it really hurts, and it gets sore, and then all of a sudden you realize you have a tongue that you don't even think about. But it also helps it push it down when you swallow. It's just to increase the friction of the food on the tongue. So the way these taste buds work is it's like a lock and key mechanism. So we have this taste bud, and if we look at the diagram here, in the bottom right corner, we see that there's the taste bud, and it comes all the way up, and at the very, very tip, there's a little kind of valley, right? That's the taste pore. And certain molecules are able to fit into that valley. Now, they're not just plain little U-shaped valleys. They're very specific shaped. They're designed to fit specific molecules. And so each taste bud is designed for a specific taste, whether it's sweet or salty or umami or I'm blanking on bitter. Am I missing one? That's it, right? Sweet, salty, bitter, and umami. I think that's it. Somebody correct me if I'm wrong. But each one is specific for each one. Each taste bud is specific for one of those sour. Yes, thank you. I'm missing sour. Okay. Then, if a molecule that our brain wants to see or taste as being one of those flavors fits into that pore, we sense it. If a sour molecule tries to fit into a salty taste bud, it's not going to work. But any molecule that fits into the sweet taste bud is going to create a sweet taste, which is how our artificial sweeteners work. And so these taste buds were evolutionarily designed to taste sugar. Sugar has calories, your body can metabolize that and get energy from it. But our artificial sweeteners, even though our body can't metabolize them and get sugar or energy out of them, they still fit into the same lock. So they go in there. The taste bud is not a smart thing. It's just a dumb little cell that when something binds, it sends a signal to the brain that the brain interprets as being sweet. And so the artificial sweeteners are designed to fit into the sweet taste bud. Next is smell. Smell is a very, very similar system to taste. We have cells that have a pore designed to fit a specific type of molecule, and when it does, it sends a signal up to the brain, and that the brain interprets as being a specific smell. And so obviously different things have very unique smells. That is the unique combination of the individual smells put together. Different ratios of one versus the other. So what you're looking at here are your sinuses. And so you don't realize it, well actually now, you may realize it now if you've had a COVID exam where they shove the giant swab up your nose, but your sinuses go way back there. So that is what you're seeing here. If you smell something, you inhale it up your nose, it comes up into your sinus, which all the way in the back behind your mouth is connected to your throat, which is why when you get a cold, the snot will run down the back of your throat. But on the very top of your sinuses is a bone. That's this ethmoid bone here. We'll learn more about that when you do the skeletal system lab. But the ethmoid bone is the bone that separates your brain up above from your sinus down below. And so we have these neurons that come directly from the brain, come down to that ethmoid bone, and they stick little fingers down through tiny little holes in the ethmoid bone. And so those neurons are actually sticking fingers down into your sinuses. So when you inhale something into your nose, the molecules will bind to those neurons that are connected directly to your brain. And so smell has the most direct route to your brain, which is why it's thought that smell is the sense that is most closely related to memories. And so you can walk into a place and smell it, and you'll immediately remember a smell to say, I was here 30 years ago, and this is what I did when I was here. Or you associate a smell with home, or you associate a smell with your grandmother's perfume, something like that. But that's because there is a very direct route to your brain. And so your brain is very well connected to the smells around you. Okay, so now we move on to the eye. So we have the eyeball itself, and we'll talk plenty about the eyeball, but we also need to talk about the other things that go with our eye. We call these accessory structures. We have our eyebrows, right? Eyebrows are up here. Primarily nowadays, they're for facial expressions. They do help prevent sweat from rolling down our forehead into our eyes, but they can only soak up so much sweat, right? It doesn't take much sweat to saturate your eyebrows, and then it just starts coming down. I don't know about you, but when I was outside at 10 o'clock this morning, apparently picking up ticks, it's Florida, it was already very hot. I started sweating quite a bit in 5 minutes that I was out there. But eyebrows do play a very important role in facial expressions. And now that I'm talking about eyebrows, you're probably looking at my eyebrows, even though you've never even noticed I had eyebrows up until now. We also have the palpebrae, which are our eyelids. Now these have a number of different purposes. Number one, it is to protect our eye. If we're going down the road, we're riding our bike or something, and there's a big cloud of dust, we kind of squint, we close our eyes at least as much as we can so that we don't get run off the road. But it protects our eyes. If something comes flying at our face, we instinctively will close our eyes. It is also to moisten our eyes. The tears on our eyes are liquid, right? Gravity wants to pull them down. And so every time we blink, we go down, the eyelids grab some water and pull them back up. So it keeps our eyes moist. And they also block light. And so when we want to sleep, we close our eyes to block that light. If we go from a dark space to a light space very quickly, while our eyes are adjusting to that change in light intensity, we're going to squint, right? And part of squinting is not just the cheek, but we're going to partially close our eyelids. We have a conjunctiva. A conjunctiva is a mucous membrane that covers not just the front of the eye, but also goes up on the inside of the eyelids, and this makes mucus. And so the mucus is to create basically lubrication. We're going to talk about the tear gland next, but tear gland makes tears, which is basically just salt water. Salt water is not a very good lubricant. But if you mix salt water with mucus, now you have a good lubricant. Does the name conjunctiva sound familiar to anyone? Does it remind?
Like pink eye.
Pink eye, yes. What is the technical term for pink eye?
Like conjunctivitis.
Yes, conjunctivitis. Anything itis is an inflammation of whatever was in the word before itis. And so conjunctivitis is inflammation of the conjunctiva. Just like hepatitis is inflammation of the liver. And so conjunctivitis is generally an infection of the conjunctiva that causes it to get red and swollen and secrete all kinds of nasty crap that you have to basically chip off your eye so that you can open it. Red eye is not something that you want to get. The tear gland is technically called the lacrimal apparatus. The technical term for making tears is lacrimate. I learned this kind of trial by fire. I did not know that. And I was working in the lab one time, and I had a chemical, and I worked with chemicals all the time in the lab. And it said, warning, lacrimator. And I said, lacrimator? If it was so bad, I would know what that word meant. And so stupid me, I just started working with this chemical, and all of a sudden, it hit me, and it felt like my eyes were on fire. And I took the stuff, and I ran, and I put it in the fume hood, and I started washing my eyes out, and I said, what the heck was that? I wonder if that has to do with the word lacrimator. And I looked it up, and sure enough, a lacrimator is something that makes your eyes make tears and burn. It's like pepper spray or onions or something like that. So if you ever see anything that says warning, it's a lacrimator, do not mess with it. But the lacrimal apparatus is your tear gland. The tear gland is located up here. So you know where your tear duct is, right? That's in here at the corner by your nose. The tear gland is opposite that, diagonal. On the top of the eye, on the outside. We make tears there so that in order for them to get to the tear duct, they have to cross the entire eye. If the tear duct was here, it would just go from here to here and the outside part of the eye would never get any tears. And so it is located there so that we get tears over the entire eye. And finally, we have the extrinsic eye muscles. We're going to see these in a bit, but these are the muscles outside of your eye, which is why they're called extrinsic, think external. These are the muscles that move your eye around, that help you look in different directions. So that's what we're looking at here. So we have the eyeball. It's sitting in the eye socket, which is technically called the orbit. And we see there are these muscles attached to the outside. This is a muscle here also, but they've cut it so that it doesn't block the whole eye. So they cut it here and here. If we look at the eye itself, we are going to break it up into posterior and anterior chambers. And the border between anterior and posterior is going to be the lens. Now, we haven't talked about the lens yet, but you know we have a lens in our eye. And so when we talk about the front and the back of the eye, the lens is the dividing point. The anterior part of the eye, called the anterior cavity or the anterior chamber, is in front of the lens, and the posterior cavity or the posterior chamber is behind the lens. If we look at the walls of the eye, we're going to have three distinct layers, and these are going to have their own slide. But it is the fibrous tunic, the vascular tunic, and the neural tunic. So these are those extrinsic muscles. And so the diagram on the left is the same thing we were looking at before. I want you to look at the one on the right. Now I want you to imagine how we can use these muscles in order to move the eyeball. So look at this medial rectus muscle. When muscles contract, they shorten, okay? And so when they shorten, it is going to pull this end of that muscle towards wherever, or this end of that muscle towards wherever the other end is anchored. And so, which way is that going to make the eyeball look? On the slide, is it going to make it look to the left or make it look to the right if the medial rectus muscle contracts? So somebody said left. So in this situation, if this contracts, it's actually going to pull this part of the eye that way. Because one end of that muscle has to stay attached to the eye. The other end has to stay connected to a bone somewhere over here. And so the only way for that muscle to get shorter, but not rip off one of the ends, is for this part of the eye to get closer to that bone. And so if this muscle contracts, it's going to pull the eye and make it look to the right. If this one contracts, it's going to make it look left. This one makes it look up. This one makes it look down. By combining movements of those four muscles, you can make the eyeball look any direction. If these two contract, it's going to look up at a 45-degree angle in between them. And so all you have to do is make different combinations of them in order to make them look any direction that you want the eyeball to look. Now we get back to those tunics. Remember I said the tunics were the layers of the eyeball. So the eyeball is, for the most part, think of it like a water balloon, okay? Most of it is full of water or jelly. You have kind of the wall around the outside. At the very front, we do have some special structures that we'll talk about. But for the most part, it is the outside. So if you look at that, there are three layers. You have the fibrous tunic, the vascular tunic, and the sensory tunic. That other slide called it the neural tunic. Those are interchangeable terms. The fibrous tunic is the outermost layer of the eyeball. Fibrous should bring to mind fiber, that is tough, that is for protection. So this includes the sclera, which is the white of your eyes, and the cornea. The cornea is right in the very front. It is clear. So the sclera is white, the cornea is clear, but they are one tunic that goes around the entire eyeball. It's just in the front, it is clear. And so because it's different, we gave it a different name, which is cornea. When we move inwards from the fibrous tunic, there's a second layer underneath that fibrous tunic. It is made up of the choroid, the ciliary body, and the iris. You're familiar with the iris. That's the colored part of your eye. It expands and contracts to make your pupil larger or smaller, depending on how much light is coming into our eyeball. The choroid is a kind of just a general extra protective layer. It's underneath the sclera. So it is not going to be in the front. It's not behind the cornea. It's only behind the sclera. And it doesn't have really any specific purpose other than being a second kind of structural layer. The ciliary body, however, which is kind of right around the iris, is going to be a muscle. This is going to be an intrinsic muscle inside of the eye that is going to pull on the lens in different directions, changing its shape, allowing us to focus at different distances. Finally, we have the sensory or neural tunic. This is the retina. So the only thing you need to worry about, So there's a deer walking by with the tiniest little fawn out here. It must be an absolute newborn. I wish you could see it, but you would never see it. But back to the retina. So the retina is in the very back of your eye. It is pigmented, meaning it has color. Where do you ever see the colored retina in an eyeball? You can't look at my eyes right now and see it, but I guarantee you there's been situations where you have seen the colored retina in the back of someone's eyeball. Can anybody think of it?
Like when you flash light on it?
Yeah. The red eye, when you take a picture with a camera. What you're seeing is light bouncing off of the retina and coming back out. Or if you're driving down the road and you see a pair of deer eyes, green, looking back at you. So different animals have a slightly different colored retina, and so you can might be able to tell what animal you're shining a light on based on the color. In this retina is a neural layer that has the photoreceptors, and we'll learn all about those photoreceptors. So, which of the three tunics was the retina in?
C.
Yes, retina was in the sensory tunic. What was included in the vascular tunic?
The iris.
The iris, it's one.
The ciliary body.
Ciliary body. There was another one that started with a C.
The choroid.
The choroid. Yep. Fibrous tunic is made up of what?
Sclera and cornea.
Sclera and cornea. Very good. Someone asked, is the color of the retina normally red on humans? And the answer is yes. I can't think of a picture. I mean, people generally have red eye in pictures. I Other animals tend to be green. I don't normally see green in humans. I'm guessing if you ever see a different color in a picture, it's probably about an angle or the light that was shined rather than the difference in the retina. So now we're going to have light coming into our eyeball. And the purpose of all of those things in the eye is to focus the light onto the retina. We need to be able to focus at different distances so that we can see clearly. When you wear glasses, it's because these parts of our eyeball are not working properly. So we're not focusing the light properly on the retina. The retina is basically like a projector screen. If you have a projector, when you set it up in front of the screen, you always have to adjust the lens in the front, right? In order to get it to focus. That is what your eyes are constantly doing. The first thing the light hits as it comes in is the cornea. Now we tend to think about the lens as being the thing that focuses the light. But in reality, the cornea actually does most of the focusing. Now, we can't change the shape of that cornea. And so it's a kind of a one-size-fits-all thing. But it's going to take light from lots of different angles, grab onto it, and shoot it towards the back of the eyeball. So it acts as, kind of as a rough focus. If you've ever used a microscope or maybe a telescope or binoculars, you might have had two different focus knobs. One is the rough focus to get you close to the right point, and then you have a fine focus that gets it just right. So the cornea is going to be the rough focus. It gets it pretty darn close. So the light comes through the cornea, it gets focused once, it then goes through what's called the aqueous humor. It says a liquid, it's more like a clear jello. It is in the anterior part of the eye, so in front of the lens. So this is what's filling the eyeball in front of the lens. This is the aqueous humor. It is going to bend the light a little bit. When we bend the light, we call that refraction. Refraction is the bending of light as opposed to reflection, which is the bouncing of light. So here we're going to take light coming in and bend it to focus it on one specific part, which is the retina in the back of our eye. Then we go through the lens, which is the fine focus. We pull on it with the ciliary body to get it just the right shape, depending on how far away we're trying to focus. Then we go through the vitreous humor, which is the liquid or the jelly that's in the back of the eye. The vitreous humor is going to take up the bulk of the volume of the eye. The part behind the lens in our eye is much bigger than the part in the front, and it is filling up, or it is filled with the vitreous humor. So if I go back, let me find diagram. Yeah, so here. So imagine the light coming into this eyeball. So it's going to come from the right, it's going to hit the cornea, it's going to get focused so that it goes through the pupil. The pupil, though, is not empty space. There's aqueous humor there with that clear jelly. Then it goes through the lens. After it passes through the lens, it goes through all of this vitreous body and then projects on the retina in the back. Once that light hits the retina, we need to convert that. It is currently light, which is photons. Little packets of energy flying through the air. It hits that retina in the back, but we need to send that signal to the brain. And the brain doesn't accept light as a signal. It receives electrical signals. And so what's going to happen is when it hits the retina, we're going to have cells in the retina that when they get hit by a photon, it causes ion channels to open and causes an action potential to occur, which then sends a signal to the brain that our brain interprets as an image. That's what you're seeing when you look around. You're seeing your brain's interpretation of a series of electrical signals that were triggered by light hitting photoreceptors. Now, we'll talk more about these different cells on a different slide coming up. So it hits the back of the eyeball, and then it gets converted into the electrical signal, and it goes through the optic nerve to the brain. Now, there's a couple things I want to point out here. And there are terms here. I'm not too concerned about the terms. I'm more concerned about the concepts that this diagram shows. Notice that there are two parts to this optic nerve for each eyeball. There is part that goes to the left side, part that goes to the right side for each eye. Not only that, but the parts of the nerve that are on the outside, the lateral portion of that eye, are connected to the brain on the same side. The part of the cranial nerve that is attached to the eyeball on the medial part of the eyeball actually cross over, and it crosses over at this optic chiasma, or the optic chiasm, sometimes people leave off the A, that crosses. And so it's actually connected to the other side of the brain. And so part of my right eye sends signals to the right part of my brain, and the other part of my right eye sends signals to the left side of my brain. Why do you think that might be? That seems like it's getting very complicated, but there must be a reason. So depth perception, I can see that maybe helping, but depth perception has more to do with comparing one eye to the other rather than comparing the two sides of one eye to each other. So imagine an eyeball here. We're going to focus on the one on the left, and you can see the pupil here, right? So if light is going to come into that eye, and it's going to hit the part of the retina on the right side, what direction is that light going to come from? From the left, from straight forward, or on the right? From the left, yes. And so it's going to come in at an angle like this, go through the... You can see my pointer, correct?
Yes.
Okay, good. So the light's going to come in from the left, like this, and hit the right side of the eyeball. Now, imagine something is flying at me from the left. What is my natural reaction going to do? Block it. So if something is flying at me from the left, which hand am I going to try to block it with? My left hand. What part of my brain controls my left hand? No.
The right side.
The right side. And so, if something hits the right side of my left eye, that means that the light is coming from my left, and so I need to react with my left hand, which is controlled by my right side of my brain, and so I'm going to send the signal from that part of the eyeball to the right side of the brain. Same thing with the inside of my right eyeball. If it hits over here, that means the light is coming from here, which I'm going to react with my right hand, which is controlled by my left brain. If it hits on the outside of the eyeball, that means that it came from the opposite direction over here, which means I'm going to react with my left hand, which is controlled by my right brain, And so the right side of my right eye is connected to the right side of my brain. Does that make your brain explode? Just thinking about that.
Well, it makes sense because the right ones are on the right side of the eye and the left ones are on the left side of the eye on both of them.
Yes. And so it makes logical sense, but you need to be able to walk your way through that logic. And so I will not ask you, and you will not be asked a question like, if light comes from the left and enters the right eye, what side of the brain will receive that signal? That is not a type of question that you are going to receive. But you could get asked a question that relies on the basic principle of light crossing over, where sometimes your left eye is sending signals to the right side of your brain. But it would be a very generic, general question. So what is sending that signal? It's being sent by our rods and cones. Oh, I think it's a good time for a break. Look, my phone says it's 6:52. We'll come back at 7 o'clock, which is 8 minutes from now. Give myself a tick check here. That's what happens when you insist on walking around in Florida in shorts. So I'm sure you've all been dying to know what's going on with the boat. Whether you want to know or not, you're going to hear it. If you don't, you can leave. But the boat is now back in the water, which is good news. And I actually figured out what sank it. And so I used science to unsink it, but it turned out that it was science that sunk it in the first place. And so when I pump rain out of the boat, I have a bilge pump. It's not built in or anything. It's just a pump that sits in the bottom of the boat, and I have a hose attached to it. And when I want to pump water out of the boat, I throw the end of the hose over the side of the boat. I plug the pump in, and it runs. Apparently, that night, before it sank, after we got a whole bunch of rain, I went out and I pumped it out. And when I finished pumping it, I apparently didn't bring the hose back into the boat. And when I shut the pump off, the water that was in the hose going up over the side of the boat got pulled back down by gravity and started a siphon. And I siphoned the entire lake into my boat until it sank. Which is bad news because it caused the boat to sink. But it's good news because there's nothing to fix. All I have to do is not leave the hose hanging over the side of the boat and it won't sink again. So that is the end of that saga. Okay, so it is 7 o'clock. So we're going to pick up where we just left off. So we're talking about light hitting the back of the eyeball and a signal being sent to the brain. But we need to convert that light into an electrical signal, that action potential. And that is going to happen and be caused by some very specialized cells containing very specialized proteins. Now, you've probably heard of these cells. These are the rods and the cones. So the rods are for our night vision. This helps us to see in low light conditions. It's also what we use for our peripheral vision. It is not very good at, or they are not very good at producing clear images, which is why when we see something out of the corner of our eye in our peripheral vision, it's kind of blurry. It has less to do with the focusing, and it has more to do with the fact that it's being seen by the rods rather than the cones. The rods are black and white only. They do not see color. Rods are the more common or the more numerous sensing cell in our or on our retina. They are only on the outside of our eye, of our retina, so around the outside. They're not in the center, and the specialized protein that the rods call or contain is called rhodopsin. Now, rhodopsin has an H in there, but it is a silent H. And so the way I remember that rods have rhodopsin is that it looks like rod, right? And so you can remember that rods have rhodopsin. Rhodopsin is basically an ion channel that when it gets hit by a photon of light, pops open. When it pops open, the sodium comes rushing in, we get the action potential, yada, 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 and we're able to see whatever it is that we are seeing. In addition to the rods, we have cones. Cones are only active during the day or during bright light conditions. They allow us to see very, very sharply. They give us a nice clear picture. These give us our color, but we do not have nearly as many cones as we have rods. And they are in the center of our retina, in an area called the fovea centralis. That is the very center part of our retina. This is a diagram showing our rods and our cones. And they get their name because they are shaped like rods and cones. And so we look at the top diagram, we see there's these green cells. These are the rods. And there's the blue cells. They kind of look like squid, but the tip is cone-shaped. So those are our cones. And so there are other cells down here, bipolar cells and ganglion cells, that also play roles in the conversion of light to action potentials. But we are not going to talk about them. I suggest that you know that bipolar cells and ganglion cells are... Boy, am I reading that right? That's really small. Yeah, ganglion cells. I'm not going crazy. That's what I thought it was. So, I would know that they exist. We are not going to cover what they do, other than they help the rods and cones convert the light into action potentials. So, where are those rods and cones located in our eye?
See the retina.
Yeah. So the light comes in through the iris, which controls the size of the pupil, then goes through the lens, which the lens is controlled by the ciliary body, and then ends up on the retina in the back, which is just a projection screen where we convert that light into nerve signals. So now we're going to move on to the ear. Are there any questions about the eye before we move on to the ear? Okay. So, to me, the ear is easier to understand how it works. It's more mechanical to it. You can look at it and say, okay, this moves, which causes that to move. It's like looking at an engine, and if you don't know how an engine works, you can say, well, I know this part moves, and if this part moves, that causes that to move, which causes that gear to move, which causes that, and eventually the wheels spin. As opposed to looking at a circuit board and trying to figure out how a circuit board works. Nothing moves. It's just magic, basically. In my mind, the eye is like the circuit board. It's all electrical signals. It's hard to see how that all happens. But the ear is more mechanical and just makes logical sense to me. So the ear has two special senses involved with it. Obviously, there is hearing, but there's also equilibrium. Our ears are involved with us keeping our balance and feeling how our body is moving. But they are both part of the ear. When we look at the ear, we break it into three parts. There is the outer ear, which contains the auricle, and this bullet point I believe I added today, and so your copy may not have the auricle included here. But the auricle is this part. It's the part that sticks out away from your head. It includes both this part and this part. So the part of the ear that sticks out away from your skull is your auricle. Once you move in from there, we have the external auditory canal. That's where you stick the Q-tips. The Q-tip package says, do not stick it down into your ear, but that is what everyone buys Q-tips for. Nobody uses Q-tips out here like the instructions say. Everybody sticks it down there until it hurts, right? That is the external auditory canal. At the end of the external auditory canal is the tympanic membrane, which is your eardrum. The eardrum is what's going to convert sound waves into actual mechanical movement. And we'll see that in a diagram, probably on the next slide. Then we're going to move in beyond the eardrum to the middle ear. The middle ear is between the eardrum and the cochlea. It's going to be an open area between those two structures. It contains the eustachian tube, which is also called the auditory tube. This is actually going to connect the middle ear down to your throat. That's going to allow two different things. It's going to allow fluid that builds up in your middle ear to drain down. And it's also going to allow pressure to equalize in your middle ear. We'll talk about both of those things when we see the diagram on the next slide. There are ossicles. Ossicles are actually the smallest bones in your body. There are 3 of them in each ear. They're going to actually take the vibrations of the eardrum and transfer that movement to the cochlea. Also included in the middle ear is something called the oval window. That is where the ossicles attach to the cochlea. Then we get to the inner ear. The inner ear is made up of the cochlea, which is where we're going to have neurons that are going to take this movement and send the signal to our brain, where the sound is detected. It's also where the equilibrium sense is going to be. So let's look at this diagram again. So we have the auricle outside. We have the external auditory canal coming in, And then at the end of that, we have the tympanic membrane, which is the eardrum. Now, when remember, sound vibrations are just literally vibrations coming through the air. They are physical vibrations. They're going to come in through the air and they're going to hit that eardrum. When they hit that eardrum, they're going to make the eardrum shake. When that eardrum shakes, it is going to push on the malleus. The malleus is one of those three ossicles. The three ossicles are the malleus, the incus, and the stapes. They're kind of like a stick, basically, connecting the eardrum to the cochlea. Imagine you have, let's say, a trampoline. Okay? If you have a trampoline, that's like your eardrum. And if you put a post on that trampoline, and then you jump on the trampoline next to that post, what's the post going to do? It's going to go up and down like this. And so if you had something up above that post detecting every time that post came up and hit it, that's essentially what your cochlea is going to do. Vibrations hit the eardrum, cause the eardrum to move. When the eardrum moves, the ossicles move, and the cochlea is going to record the movement of those ossicles. Also in the middle ear is the eustachian tube. Now, it's draining down into your throat. It has no function directly in hearing or equilibrium. It is a maintenance system. I don't know about you, but when I was little, I had tubes put into my ears. And that's because ear infections are the most common cause of doctor visits for small children. And that is because their eustachian tubes are not properly developed yet. This doesn't mean they're not going to develop, it's just they're not developing fast enough. So this eustachian tube is supposed to be able to open and close as needed. If we need to drain the fluid from the middle ear down to get rid of it, it should open. But in young kids, it doesn't like to open. And if it doesn't open, that fluid builds up, and bacteria like to grow in wet, dark environments. And so we get middle ear infections. Another function of the eustachian tube is to equalize pressure. Before this all COVID crap hit the fan, we like to get on airplanes. And when you get on an airplane and you start going up and you start to go down, you feel the pressure in your ears. And so you have to try to pop your ears, right? When you pop your ears, what you're trying to do is force that eustachian tube open. When you go up in an airplane, what happens to the atmospheric pressure around you? Or when you go up on a mountain, same thing happens when you go up a mountain.
It increases.
Did you say increases or decreases?
Increases.
As you go up, atmospheric pressure is actually going to decrease. So higher you go up in the atmosphere, pressure decreases. And so the pressure on the in the external auditory canal, on the outside of the eardrum, is going down. In the inner ear, it's kind of an isolated environment. And so its pressure is going to stay the same. If you have the same amount of pressure pushing outwards on the eardrum, but less pressure pushing in on the eardrum, what's going to happen to the eardrum? Less pressure pushing in, same amount of pressure pushing out. What happens with in air? If you've ever had a balloon and you've gone up a mountain or gone down a mountain, have you ever seen what happens? Or if you have a capped water bottle, yeah, somebody says it's going to flex outward. That's right. We have the normal amount of force pushing outwards, but less force pushing inwards. And so that eardrum is actually going to get pushed physically outward, which is painful. That's that feeling that you get. And so you start to try to pop your ears. And if something happens for one reason or another, you're not able to, it gets very, very painful. Which is one of the many reasons that little kids, and particularly babies, will cry on airplanes. Because their ears aren't equalizing their pressure. And they're not going to do the whole hold your nose and blow thing. You can't give them gum. Chewing likes to cause that eustachian tube to pop open. So they can't equalize their pressure, and it gets very, very painful. And so when you blow on your nose, when you hold your nose and blow, what you're doing is you're putting very high amounts of pressure on the bottom part of that eustachian tube, and that high amount of pressure causes that eustachian tube to pop open, and when it pops open, everything equalizes, and then you're good to go. So, coming back to the sound, we take the vibrations of our eardrum, take it through the ossicles, and we're going to take it to the cochlea here, and we'll see the cochlea, how that works in just a bit. And so, remember this path of sound. We're going to go through the auricle, the outside part, through the external auditory canal, to the tympanic membrane, which is the eardrum. Then we enter the middle ear, which it goes through the ossicles, the malleus, the incus, and the stapes. The stapes, which is one of those three ossicle bones, is connected to the oval window on the cochlea. The cochlea is in the inner ear. Now, how this works is a little bit harder to imagine. Have you all seen a syringe? Have you ever played with a syringe without a needle on it? A syringe without a needle on it. If you put your finger on the end and you push on it, you can feel the pressure, right? When you start to push it in, you feel pressure. It gets harder and harder to push, and then when you let go of it, the pressure pops the plunger back out. That's the same thing that's going on here. The stapes is essentially like a plunger pushing on the cochlea. The cochlea is filled with fluid. When you push in on that oval window, you create an increase in pressure, and that increase in pressure will travel through that kind of snail-shaped, spiral-shaped cochlea. And it goes, that kind of pressure wave will go through it to the very center, where we have little hairs that are going to sense that vibration and send the signal up to the brain. So what was the name of the ossicle that is connected to the oval window, which is on the cochlea? Yep. Stapes is the third one. It is the one that is connected to the cochlea. So these are those sound waves. So remember, sound waves are vibrations in the air. They're going to come in, get converted from sound waves in air, get converted from air vibrations to eardrum vibrations, which causes the ossicles to move. But the ossicles are connected to the cochlea, which creates pressure waves in the cochlea, which are then going to get sensed by little hairs. Yeah, here we have it. So the hairs are in what we call the organ of Corti. The organ of Corti is the part of the cochlea that actually senses the sound. So do I have? Yeah. So what you're looking at the cochlea. It looks like the snail here. We've taken that and we've cut it in half. Now, when I looked at this diagram, I realized they did not draw it properly. When they draw this little box here, they think that they're showing it. Seemingly, they're trying to tell you that what you're looking at here is what's inside that box, but that is not true. what you're looking at is an entire cross section of this cochlea. Just cut it open and you're looking inside. So in the center of it is this organ of Corti. And if we zoom in on that organ of Corti, oh, by the way, down here in the words here, they're telling you some different parts of the cochlea, like the scala tympani. You do not need to know those words. So this is just kind of a visual slide. Don't worry about what's in the words at the bottom here. Now if we zoom in even further, we see these hair cells. So the hair cells have these hairs sticking out, and they are attached to this little membrane. The name of that membrane is not important to you. I need you to know that there are hair cells, and those hair cells have hairs that when those pressure waves reach the organ of Corti, it's going to cause those hairs to vibrate. And when those hairs vibrate, those hairs are actually attached to ion channels. When the hair vibrates, it causes the ion channel to open up. The ion channel opens up, sodium rushes in, we get an action potential, and the magic happens. That is how we convert pressure waves, which started as waves in the air, into an action potential. Now the last thing to talk about is equilibrium. Do you have questions on hearing before we move on to equilibrium? Okay. So we have a vestibule located within our ear. This is what it's going to monitor our equilibrium. It is, so it is connected to the cochlea. It is, if you look at the cochlea, we have these little loops up here, and then we have a vestibule down here, which is made up of the macula and the utricle and the saccule. And so the vestibule is going to measure the equilibrium. And we talk about two distinct types of equilibrium. We have static equilibrium and dynamic equilibrium. Static always refers to stationary. That's what static means. Like static electricity is not moving through a wire. It is staying in one place. So static equilibrium is our balance, essentially. We have balance if I am just sitting here or if I'm standing here. It doesn't matter whether I'm moving or not. I am aware of how I am tilted, right? I know that I am sitting upright. If I close my eyes and all of a sudden the chair tilts back, I am going to know that I am tilting back because of this static equilibrium. And the static equilibrium is measured within the saccule and the utricle. The other part of equilibrium is dynamic equilibrium. Dynamic equilibrium is feeling of acceleration. Like when you hit the gas pedal in your car, or you hit the brake in your car, or you turn sharply in your car. You know that you are accelerating, going faster. Deceleration is technically negative acceleration, so that's slowing down. Turning is also, based on the physics term acceleration, considered acceleration. Acceleration does not have to involve a change in speed. It can be also a change in direction. So that is going to be carried out by the three semicircular canals. We'll talk more about each of these types of equilibrium separately. So this is the static equilibrium. Okay? So this is what's going to tell us how we are oriented in space. Specifically, it is going to tell us how our head is oriented. Now, if we keep our head in line with our body, we know that's the case. And so if our head is tilted and we know our head is in line with our body, we know that our body must be tilted. But if we know that we're tilting our head either way, we know that our head must be tilting. Now, I have taught this class for years and years, and every time I do this lecture, I always forget to bring the visual aid. But now that I'm teaching it from home, when I realized I forgot, all I had to do was go into the kitchen and grab my visual aid. So this is going to show how the static equilibrium works. So this is just a little bowl. It's round, essentially, in the bottom. And I have a little bead, okay? So I'm going to put the bead in the bottom of the bowl. And if I hold the bowl upright, without even looking in this bowl, you can tell me where in the bowl that bead is. Where is that bead? Is it on one of the sides? Is it in the center? Is it on the top? Is it in the bottom? Where is it?
Bottom.
It's in the bottom and in the center. So right now, assume this bowl is perfectly round. It's not. It's got a little flat spot in the bottom. But we're going to assume it's perfectly round. So it's going to settle in the very bottom of it. And so essentially we're going to measure static equilibrium by measuring where in the bowl the bead is. If the bead is in the center at the bottom, we know that the bowl must be upright. If I take this bowl and I tilt it towards you, where is that bead going to go? Let's see if I can tilt it far enough that you can see it, right? So where did it go? It went towards the bottom, or towards this side, actually. And so we know that if the bead is touching this side of the bowl must have tilted this way. Now imagine this bowl is in my ear. If the bowl tilts this way, that means my head must have tilted that way. If I tilt my head this way, the bead's going to roll over here. Here. I was looking at the camera and did everything backwards. So it is going to roll here. If I tilt it this way, now it rolls out here, right? And so all we have to do is monitor where in that bowl that bead is, and we know which way the bowl is tilting. That's essentially how this is going to work. We don't have a bead that is free floating around like that in there. Instead, the bead, which is called an otolith, is attached to a hair cell. And so when the bead wants to move, it bends that hair cell one way. I can't bend my finger backwards, but it's going to bend the hair one way or the other. And by monitoring which way that hair bends, we know which way the head is tilting. The harder one to imagine, and I can't give you a visual aid. I'll give you kind of something to imagine. But the harder one is dynamic equilibrium. This is going to tell us that we are moving in one direction or another. This could be the whole body moving like in a car, but it's also the head moving around the neck. And so this is measured by semicircular canals. Now, looking at the diagram, and it was on another slide, how many semicircular canals are there?
Four.
Three. There's three.
Three.
There's three.
No.
It's hard to, you don't really know what you're looking at unless you know what you're looking at. But there are three semicircular canals there. Thinking about space around us, how many dimensions are there? How many dimensions are there? Don't overthink this.
Three.
There's three dimensions, right? There is left, right, up, down, forwards, and back. If you're talking about a graph, we have the X, Y, and Z axes. And so essentially that is why we have 3 semicircular canals. Each one of them is going to measure movement in one axis. One of them is going to measure movement up and down, one measures movement side to side, and the other measures movement forwards and backwards. Now, if we move diagonally in any direction, we're going to activate 2 or 3 of those. But we get different combinations of activation, and we know exactly what direction we are moving. Now, the way this works is, again, we have hair cells, that's hair, that are going to detect, are they flopping this way or are they flopping that way? Now, what I want you to do is I want you to imagine a garden hose. And that garden hose is going to be like one of our semicircular canals. Now, imagine in that garden hose we have water. Okay? So you have a garden hose laying out in the yard, you run some water through it, and you turn the hose off. There's always a little bit of water left in that hose, right? Now imagine you have the hose laid out in the yard. The hose is turned off, but there's still some water in the hose. If you grab the hose at the house end and you yank it really fast, what's going to happen with the water in the hose?
It'll go out.
Yes, it'll move, it'll come out the end of the hose. Now, in physics, remember, everything is relative. So she said, and my initial reaction was also, the water will come, well, she said the water will come out. And my initial reaction was, the water will move towards the end of the hose. But that is not true. The water is not moving towards the end of the hose. The end of the hose is moving. The water is trying to stay in one place. The hose is moving around the water. Now, imagine we had little hairs inside that garden hose. Okay? So we have a garden hose running sideways like this. The hairs are attached to the garden hose. If I yank the hose this way, the water is going to want to stay in place. And so it's, well, let me do this. Let me put the hair that way so that my joints work the right way. If I try to move it this way, the water is going to force that hair to bend over. The water is going to put force on that hair, and it's going to bend it over. If I move the hose the other way, it's going to cause the hair to bend the other way. That's how these semicircular canals work. They are filled with fluid. If we accelerate one way or the other, it causes the hose, the canal around that fluid, to move, which causes the water, which wants to stay in place, to push on those hairs and cause the hairs to bend. Depending on which direction those hairs bend, we know that we're accelerating this way or this way in that axis. Now, I understand that's much more complicated than the bead in the bowl, but are you able to imagine how this works? I will assume that no news is good news. And the good news is that that is the end of the lecture. So are there questions about hearing, about the ears, about the eyes, taste, smell, anything from today?
I'm wondering if, you know, people who have, like, say they have a sweet tooth or they prefer certain things, I wonder if they have more of the sweet.
They could. I don't know the answer to this. My guess would be it has more to do with the brain interpreting it as a more pleasurable experience. That would be my guess. Like I said, I don't know what it is. So somebody said, what is vertigo? So vertigo is a loss of equilibrium, right? And so a couple of different things can cause that. So vertigo has no one specific cause. It's essentially going to cause this static equilibrium mechanism to stop working. And so people will think they are falling over when they're actually standing up. And when they're trying to stand up, they'll fall over because when they're standing up, their body is telling their head that they're actually falling over. And so their muscles do weird things and they fall. Usually, what happens is if somebody gets an inner ear infection, that inner ear gets inflamed. Anytime something gets inflamed, it expands and it's going to squeeze this whole system. When it gets squeezed, nothing works properly. So that's going to be something kind of like a temporary vertigo. If that infection stays around long enough, that inflammation will actually damage this mechanism, and then you can have permanent vertigo. But vertigo is going to be some sort of a malfunction of the static equilibrium. Other questions? Okay, so that is the end of this lecture. Now we do have an exam due next week on this lecture, last week's lecture, which was the autonomic nervous system, and the week before that, which was the central nervous system. So we have time is it? 7:30. We can do some review. I'm not going to force you to stay for a review. Obviously, I can't force you. All you have to do is click leave and you're gone anyways. But I'm not going to tell you you need to stay. And so what I'm going to say is if you want to leave, go ahead and leave now. If you want to stay and do some review, I think what we'll do is I'll ask if you have specific questions. If you don't have questions, we'll go probably through the learning objectives, and the final exam study guide is kind of a checklist, and go through them step by step. So if you feel like you're good to go, you're free to leave. If you want to stay for study, we can do that. And so we'll take a 7:37. We'll take a 3-minute break right now. If you want to leave, leave. So somebody said, will this be posted on Canvas? I'm going to continue recording, and so the recording that goes up on Canvas will include the review. If everyone leaves, though, I'm not going to stay and do the review by myself. So that's kind of one catch to the system.
I'm here.
Okay. So one person is going to bite the bullet, take one for the team, stick around, and learn something. Okay, so it looks like most people left. And so the three of you that are here, do you want a break or do you want to just continue?
Yeah, I'm fine to continue if that
Okay.
works for everyone else.
Okay, so everybody said continue. So let's just go. So before I start blabbering, do you have specific questions that you want to ask?
I've noticed that every exam has a chapter that's a little bit heavier questions on. Like the first one was more heavily on like mitosis and
Because I mean, I purposely try to make equal numbers of questions from each lecture. Maybe unintentionally, I focus more on one topic versus another, but that is completely unintentional, and I'm not even aware I'm doing it.
Maybe I just think it's a harder question.
No, because I make a definite point of trying to put equal numbers of questions from each lecture.
Okay.
So I'm trying to pull up the other lectures. While that's going on, let's look at the learning objectives. This poor old computer. Come on. Muscle, nervous system. So someone asked, should we memorize all of the cranial nerves and all of the structures of the brain intensively? For the cranial nerves, the only one that I will ask you specifically about name and number is the vagus nerve. The others, don't worry too much about. Or I said will ask you. Can ask you is a better way to put that. I will not ask you what is the name of cranial nerve number 3. For the structures of Vegas is, now you got me question. Vegas is 10, isn't it?
Okay, I don't know why I thought of that number. I don't know why.
Now I gotta look it up.
I'll figure it out.
I'm pretty sure it's 10. Yeah, it's 10, which is an X.
Got it. Thank you.
Okay, so this one starts with the central nervous system. Oh, getting back to the other part of your question. Structures of the brain. So we had lots of different structures of the brain at different levels. I think for the most part, structures of the brain are probably something you need to know. I'm just spitting some out. I mean, we had the cerebellum and the diencephalon, which included the thalamus and the hypothalamus, and the epithalamus, which is the pineal gland. And we had the parts of the brain stem, and we had the cerebrum. What else did we have? Things like that you definitely want to know. And the meninges, things like that you definitely are going to want to know. Okay, so central nervous system. Number 1 says, give an overview of the major regions of the brain. That's what we just did, essentially. Differentiate gray matter and white matter in terms of structure and function. What's the difference in structure between gray matter and white matter?
Myelination.
Yes? Which one has myelin?
White matter?
Yes, because myelin is white, and so white matter is white because it has myelin. Now for function, what is the difference in function between gray matter and white matter? Remember, white matter is white because it has myelin. What's the purpose of myelin?
To protect or to...
Yep, to protect. What part of the neuron is myelin wrapped around? The axon. In order to speed up signals. And so white matter is for carrying signals from one location to another. Gray matter is where that signal is getting carried to. It's the end of the line. So it doesn't have axons. So there's no axons, there's no myelin. We don't need myelin to speed up the signal because the signal's not going anywhere once it gets there.
So it's like the white matter parts will feed into the gray matter parts.
Yes. Yep. Number 2, what are the lobes of the cerebrum? What's this one? Frontal. What's this one in the back? All the way in the back.
No, occipital.
Occipital is all the way in the back. Bald spot lobe is parietal. These are temporal. Yep. What is the name of the fissure that separates the 2 hemispheres of the brain? Yep. Go. Yep. Longitudinal. Longitudinal. Where is the primary? Boy. Where was the primary motor area? Do you remember?
That's the precentrical.
So motor was front.
Oh.
Motor was front. Where is that slide? Where? Yeah. So it's the frontal lobe, but it's not the very front, and so it's still the frontal lobe because this is the dividing line between the frontal lobe and the parietal lobe. So the motor area is in the frontal lobe. The sensory area is the parietal lobe. Visual was in the back. What are the ventricles? What are their names? What do they do?
Oh, lateral, third.
Yeah, you have the two lateral ventricles, one on each side, and then we have the third and the fourth. What is their purpose? They're reservoirs. Good. Reservoirs of cerebrospinal fluid. What makes the cerebrospinal fluid? Starts 2 words. First one starts with a C. Second one starts with a P. Choroid plexus. Choroid plexus was the. I could pick it out of a list. Yeah, it is multiple choice, and so that's good. If you're looking at a cross section of the brain, how do you find the diencephalon? Where in the brain is the diencephalon?
It's in the middle with the thalamus and stuff.
Yeah, so it's in the center, and so on a diagram, like you said, you can find the corpus callosum, and it's below the corpus callosum. What are the parts of the diencephalon?
That would be the thalamus, or the hypothalamus, and then the pineal.
Yep. So thalamus, hypothalamus, and some it can be considered the epithalamus, which is made almost entirely of the pineal gland. What is the importance of the hypothalamus?
Homeostasis.
Process nerve inputs. That sounds more like the thalamus. The thalamus takes signals coming up the spinal cord through the brainstem and then sends them out to different parts of the cerebrum. The hypothalamus does what? It maintains homeostasis by controlling the autonomic nervous system. What does the limbic system do? Learning and emotion. Yes. Learning and emotion are the limbic system. 3 major regions. What are the 3 major regions of the brainstem?
Medulla oblongata.
1. Pons and midbrain. Yeah. There you go. So we got 3. We didn't talk too much about very specific functions of each of those 3. But what is the primary function, do you remember, of the midbrain? So think about where in that brainstem is the midbrain. It's kind of three in a row. Which one is the midbrain?
The one that connects the main brain to the brainstem.
Exactly, yes. And that is the main function of it. It serves as kind of a connection point between the brainstem or the spinal cord and the cerebrum. Now let's lump the pons and the medulla oblongata together. What's kind of their function? Similar to the hypothalamus.
Like stuff that you're not thinking about.
Yeah. Autonomic nervous system.
There.
And so that's going to include, like I said, visceral functions, your organs, the cardiac center, which the primary control of your heart rate is in the medulla oblongata. Cerebellar structure. If you were to describe the structure of the cerebellum to me, how would you describe it? Tree-like. What makes it tree-like? You're correct, but what makes it tree-like? The branches of the matter. So if you cut it open, there was gray matter and white matter, and the white matter was kind of in the center, and it had these branches that went out. Do you remember the name for the branches of white matter? It's something to do with trees. Yeah. If you had a list, you could pick it out, right? It's the arbor vitae. What else can you tell me about, without cutting it open, what can you tell me about the structure of the cerebellum? How is it similar to the cerebrum? It has 2 hemispheres. So it has a left and a right hemisphere, just like the cerebrum does. What is the primary function of the cerebellum? Say it again?
Balance.
So I think you have the right idea. I'm not crazy about the word yet you're using. What's another way to put that?
Equilibrium.
No. Some very similar, but once we get it, I'll explain the difference between them. Anybody? If somebody is clumsy, how do you describe them?
Coordination.
Yeah, there you go. So the cerebellum is for coordination. It makes sure things move at the proper time. Part of the input that controls that is balance and equilibrium. Now, somebody can fall over because they have poor coordination, or maybe their coordination is fine, but their equilibrium, their system up here in their ears is not working properly. But balance and equilibrium are separate than coordination, even though they may manifest themselves in the same actions. What are the 3 meninges?
Dura, mater. Yeah, that one.
Yeah. Dura, pia, and arachnoid. What is the order of them? Starting from the Let's start from the inside.
Pia, the arachnoid, the dura.
Yeah. The dura is the outermost one. The pia is the inside one. The arachnoid is the middle one. So, I don't know if you saw me laughing. I said, what are the three meninges? And then in my head, I imagined the three meninjas. And then my head went to the three Ninja Turtles. So yes, I mentioned it before, I'm insane. So we talked, we missed one. We talked about how CSF is formed. And so where is the majority of the cerebrospinal fluid made? In the ventricles, primarily the lateral ventricles. It's made in all of the ventricles, but most of it is made in the lateral ventricles. So we have the cerebrospinal fluid in the ventricles. Where does it go after the ventricles? Cerebral aqueduct, yes, which leads down to where? The spinal cord. And where else? So we've got the inside of the brain. We've got the spinal cord. What are we missing? Back up the cranial cavity in order to go where? Into the blood. So we're going to reabsorb it into the blood, yes. So into the blood is the final destination. Up in the cranial cavity, we need to bathe the outside of the cerebrum with it also. That's where I was going with that. We talked about the cranial nerves, and I said we're just worried about number 10. Don't worry about the others. For the spinal cord, what do I want to ask about the spinal cord? If you were to describe the gross structure of the spinal cord, how would you describe it? Say it again. You're kind of quiet.
Oh, it's a line with spikes off the side?
Yes. Oh, so yeah. So you are looking at it even more zoomed out than I am, which is fine. And so you're imagining a spinal cord coming down, and then you have spinal nerves coming off the side. And that's good. Now let's take it and let's slice it. So you're looking at a cross section of it. How would you describe it? That's the wrong button.
Like a butterfly-looking thing, which is the gray matter, right?
Yes. And so we have the gray matter inside, in the middle with the butterfly shape. And then we have the white matter around it. Without going too much into specific names, we had kind of lobes of the gray matter. What was a name for the lobe?
Yeah, those are the horns, right?
They were the what?
The horns. Like
Yeah. The
Yeah.
horns. We had the I don't remember. We had the lateral horns. We had was it dorsal? Or was it posterior? Now I'm questioning him again. I think it was dorsal.
I think so too.
Yeah.
So that one calls it both. And that's the roots. Dorsal. There we go. Dorsal horn. So that's what our official name for it was. What else? Well, What is the function of the gray matter? What is the function of any gray matter? It's gray because it has no myelin, which means it is the end of the line, which means it is an integration center. So it is where some thought is going to occur. What is the purpose of the white matter then?
It carries the signals.
Yep, it carries signals up and down the spinal cord. If you were going to make a general definitions in the front or the anterior part of the spinal cord, what direction do the signals go?
In the front, they go down.
Yep. In the front, they go down because back up, which means in the front, are they sensory signals or motor signals?
Motor?
Yes, they're motor. Sensory signals go towards the brain because the brain has to interpret those senses. And motor signals come from the brain because they're going back down to the muscles. What else? What are the parts of a reflex arc? So if I put my hand in a hot stove, walk me through that process of pulling my hand back.
So you have the sensory
Yeah.
receptor,
So the receptor senses that it's hot. What has to happen next? We have to send the signal somewhere, right? And what's going to send that signal? Then there's going to be a nerve or a neuron. What kind of nerve or neuron? Is it sensory or motor? Sensory. So the sensory neuron carries it up to the spinal cord. Where does it enter the spinal cord?
The back.
Correct. The dorsal root. The spinal cord, the gray matter in the spinal cord, then acts as an integrating center. It decides, okay, I need to pull my hand back. And so it's going to send the signal back out the
anterior.
Correct. Through the motor neuron back to the effector, which in this case is a muscle causing my arm to pull back. So those are that's the reflex arc. It goes from the hand to the spinal cord and then back to the hand. Is this the last one? Yeah. What is a plexus? We had several of them going up and down the spinal cords. What was a plexus?
Those are just little hubs along the spinal cord that control certain functions in the body.
Yeah, that's a very good way of putting it. If you look at where the spinal nerves come off the spinal cord, most of them come off in just a few areas. We have a brachial plexus here with a bunch of nerves going down our hands because we have very good control of our hands, lots of senses in our hands. Then we also have plexus for our legs, right? And also up in our head. So that was the central nervous system. The autonomic nervous system, far fewer little points to talk about. So autonomic nervous system as a whole, what is its function?
It senses and reacts to things going on inside your body that you don't think about, that just kind of happen on its own.
Right. So it's going to monitor the outside world and cause our body to react to it involuntarily. That's a very good way of putting it. So we had two parts of the autonomic system. What were those two parts? Remember the angel on one shoulder, devil on the other. What were they called? Yeah, there you go. Sympathetic and parasympathetic. I honestly don't know what they mean when they say with the region. I don't. We're just going to skip that. What is the major role of the sympathetic division? Excite. It turns everything up. It turns on that fight or flight mechanism. Parasympathetic is the opposite. It is the rest and digest system. It tries to calm everything down. What neurotransmitters do the sympathetic and parasympathetic divisions use? Yeah. So we have acetylcholine and norepinephrine. Where in those divisions are each of those two used?
Acetylcholine is used by all of both systems.
Yep. So acetylcholine, so remember in the autonomic nervous system, whether it's sympathetic or parasympathetic, there's always two neurons. And so there's two locations that neurotransmitters have to be used. Between the presynaptic neuron and the postsynaptic neuron, and between the postsynaptic neuron and wherever that signal is going, the effector, whether it be a muscle or a gland. And so in the parasympathetic division, which neurotransmitter is used in each case? Acetylcholine, yes, period. Parasympathetic uses acetylcholine in both locations, always, never an exception. In the sympathetic nervous system, which neurotransmitters are used in each location? That was kind of quiet. I couldn't tell what you said.
The acetylcholine is used in the first part, and then norepinephrine is used in the second part.
Yes. So the presynaptic neuron sends its signal to the postsynaptic neuron by using acetylcholine. The postsynaptic neuron sends its signal to the effector using norepinephrine, but it can also use acetylcholine also. So in that whole situation, the only place we do not have to use acetylcholine is the postsynaptic neuron in the sympathetic nervous system sending its signal to the effector. That's the only place something other than acetylcholine sometimes gets used. What is the difference between a cholinergic and an adrenergic receptor? This one's pretty simple once you know what you're talking about. And it's one of those, you kind of hit yourself on the head when you realize you didn't think of it.
Cholinergic uses acetylcholine.
Yeah.
And then adrenergic uses adrenaline.
Yeah. So cholinergic uses acetylcholine. Adrenergic uses adrenaline, but we're not talking about adrenaline. So what was the connection there?
It's the same thing as norepinephrine.
So adrenaline is the same thing as epinephrine, but there's also noradrenaline and norepinephrine, which are equal to each other, but all four. So there's only two signaling molecules there. There's adrenaline / epinephrine and noradrenaline / norepinephrine. They both work through adrenergic receptors. So in this case, the adrenergic receptor is binding norepinephrine. For the heart, what does the sympathetic division want to do to your heart rate? Excite it. Parasympathetic wants to turn it down. Sympathetic division wants to have what effect on digestive and urinary organs?
Pause it.
Yeah, crank it down. Parasympathetic wants to turn it up. Remember, parasympathetic is rest and digest system. What about bronchial smooth muscle? So the size of your airways. What is the sympathetic division going to want to do?
Dilate them.
Dilate, which remind everybody else, what does dilate mean?
Make them bigger or relax
Yeah.
them.
Yep. Dilate means make bigger. So more airflow. What do we got? Sympathetic system on blood vessels of skeletal muscle.
So large, get larger.
Yeah. Make them bigger. We need more blood getting to the muscles so we can fight or run. Let's say blood vessels, digestive, urinary system are the same thing as what we just talked about. What was it to the skin? What effect did the sympathetic nervous system have on blood flow to the skin? Decreased it. So that if we got wounded, we lost less blood. Where are the adrenal glands?
On top of the kidneys.
On top of the kidneys. What are the two parts of the adrenal gland? Inner and outer, those are the words I like to use, but the textbook likes to use fancy words. What were the fancy words?
Medulla? No.
Medulla, yeah. Medulla was one of them.
I don't know the other one.
Other one starts with a C.
Cortex.
Yes. So we have the medulla and the cortex. Now, which one's which?
The cortex is on the outside.
Yes. Cortex is outside, medulla is inside. What do the cortex and medulla make? So, if we're looking at the slide, the outside, the cortex makes steroids, which are not involved in the autonomic nervous system. The inside, the medulla, makes adrenaline, which adrenaline is one of the catecholamines, and that is what the inside makes catecholamines, most of which is adrenaline. We kind of talked about the hypothalamus and the control of the autonomic nervous system before. So then we move on to today's stuff. What's an example of a cutaneous sensory receptor?
Like anything on your skin?
Yeah, so what's a cutaneous receptor is in your skin. What's an example of one of the types of...
Like pressure?
Say it again?
Like a pressure?
Yeah, like pressure.
Okay.
Or we also had temperature and pain, things like that. What's an example of a visceral receptor?
Internal organs, baroreceptors, and osmoreceptors.
Yeah, the baroreceptors, the pressure ones, and the osmoreceptors, the blood concentration ones. Where are our taste receptors?
On the papilla.
Right. So they are the taste buds, which are on the papilla, which are on the tongue. Where are the olfactory or the smell receptors? So if I take a big old whiff of something, where in the nasal cavity below the mucus? Yeah. So remember, we have the brain up here. I mean, if you don't remember the brain's up here at this point, you might as well. Right? You got the brain, and then you got the nerves, the cranial nerve that comes down and puts little fingers down into the nasal cavity, on the roof of the nasal cavity. So that's where the olfactory receptors were. Don't worry about the cranial nerves there.
When you sneeze, like when something makes you sneeze, is it hitting your nerve or like?
And so a sneeze it's a controlled spasm of the diaphragm. And so normally it is caused by, you get something like a tickle in here, which your brain recognizes that and says, well, there must be something in there that doesn't belong there. And so I'm going to sneeze and put this big force of air trying to force it out. You also have sun sneezes. I know that's sun sneezes are actually a genetic thing. Some people get sun sneezes, some of them don't. I do. So if I'm in the house or in the car and I go out and I take off my sunglasses and it's really bright out, I start sneezing. No one has a good explanation of what that is, other than they think it's just an overload of the brain, and the brain doesn't know what to do, and so stuff just starts going haywire, and it manifests itself as sneezing.
Wow.
What else do we have? Function and accessory structures of the eye. What were the accessory structures of the eye?
Like the eyebrow and the eyelid.
Yeah. Eyebrow, eyelid, we had the conjunctiva, we had the lacrimal apparatus.
Intrinsic eye muscle.
Intrinsic eye muscle is good. I realized afterwards that one thing that's not included on that slide are eyelashes.
Oh, right.
So what do you think the purpose of eyelashes are?
To keep things from going inside your eye as well.
Yeah, so the purpose of the eyelash is to keep things from getting in your eye. I don't know about you, but when I get something in my eye, it's usually an eyelash.
Same.
Where were we? What were the three tunics of the eyeball?
Fibrous, vascular, and sensory.
Yep. What made
Or
what?
neural.
Yeah, or neural. Sensory and neural is the same. What are the parts of the fibrous tunic?
Sclera and cornea.
Yep. Sclera is the white part. Cornea is the clear part in the front. What were the parts of the vascular tunic?
Choroid.
Choroid. Iris. One more.
Ciliary.
Ciliary body. What does the ciliary body do?
Controls the shape of the
Controls the shape of the lens in order to focus the light. What does the iris do?
controls the size of the pupils.
Controls the size of the pupils to control how much light is getting in. What are the refractory media of the eye? First of all, what is a refractory medium? What do we mean by that?
Something that will bend the light.
It bends the light. And so when light comes into my eye, what are the different refractory media that it passes through?
Cornea.
Cornea,
The aqueous
aqueous
fluid.
technically aqueous humor.
Aqueous.
Then What?
No.
Picture the eye. So you've got the aqueous humor is in front of what structure? Lens. There you go. Lens.
Okay.
So it then goes through the lens. Then what does it go through? What's behind the lens?
It's the big pocket of fluid.
Big pocket of fluid, but that big pocket of fluid will not be an answer on the exam.
I forgot what it's called.
You could pick it out of a list, right?
Maybe, actually.
There you
There
go.
we go.
Well,
That's the one.
vitreous humor. I'm not sure. I wouldn't give it too much the benefit of the doubt. I'm not sure it's virtuous, but it's vitreous humor. And then what's behind the vitreous humor? Autocorrect.
Oh.
Yeah. So, what's behind the vitreous humor?
The retina?
The retina. Yeah. So the light has now hit the retina. What converts it into a nerve signal?
The rods and cones.
Rods and cones. What's the difference between the rods and the cones?
Rods is used for night, in the nighttime or low light situations. Black and white, it's like blurry image.
Yep. So like she said, it's a nighttime, low light, black and white, blurry image. Cones are what?
Color, sharper vision.
Color, sharper vision. Rods are on the outside of the retina. Cones are in the center of the retina. Moving on to the ear. What are the parts of the outer ear?
It starts with an A. Auricle.
Auricle, yeah.
Auricle.
Auricle. Then
And the outer ear canal.
the auditory canal. Auditory canal. And the eardrum, or the tympanic membrane. The eardrum is the border between the outer ear and the middle ear, but it is technically considered as part of the outer ear. What is in the middle ear?
Ossicles.
Ossicles,
Oval window.
oval window, and eustachian tube. Yep.
There we go.
We had this question in lecture, but which ossicle is the one that attaches to the oval window?
Starts with M.
Stapes.
It's the stapes.
Oh,
The stapes. What is in the inner ear?
Vestibule.
Vestibule and
Yeah.
I think you said it, but it was kind of quiet.
Cochlea.
Cochlea. Yep. The primary function of the vestibule is what?
Equilibrium.
Equilibrium. Primary function of the cochlea is don't make it harder than it is.
Just
Balance.
versions.
Hearing. Yeah. So inside of the cochlea, what is this name of the specific area that converts the waves into nerve impulses?
The cordis or
Close.
cordis.
Corti. Oh, the organ of Corti.
Organ of cordis.
So here they call them the semilunar canals. Semilunar canals are the same as the semicircular canals. What do the semicircular canals measure?
But where you were turning your head.
So that's the dynamic equilibrium, the movement, the acceleration. So that means the rest of it is measuring the static equilibrium, the tilting, how you're oriented in space. Okay. Oh, we got more. Yeah, those aren't important. 9 and we already basically talked about. Now, when you work through outer, middle, and inner ear, you've already worked through the tracing the sound conduction, and I'm not worried about lobe of the brain where it's received. So I think, do you want to go through the study guide, or do you want to leave it as is? A lot of it would be repetition. I don't remember how much new is in there.
Quick question about the lens. You said it was like the dividing between the posterior and the anterior of the eye.
Mm-hmm.
Is it technically, like you're saying about the eardrum, is it technically a part of the posterior or the anterior?
At least I have never really seen people make such a hard definition about anterior and posterior part of the ear or of the eye like they do with the ear. And so I don't know the answer to your question. I think the reason I don't know the answer is because no one really cares about that.
Okay. Yeah, I think we're probably good then if it's just getting a lot of repetition.
Yeah. I mean, you can go through it. I think we'll just leave it at this. If you have look through the study guide, if you have questions about it, just send me a message and we'll figure it out.
Thank you for the review.
I hope it helps. Good luck on your exam. If you have questions, let me know.
Thank you.
Thank you so much.
See you next Tuesday.